For decades, the cosmetics industry has harvested natural plant metabolites, like kojic acid, to brighten our skin. We extract these chemicals from nature, bottle them up, and apply them externally. Synthetic biology approaches this relationship from the opposite direction. Instead of pulling chemical compounds out of plants, scientists are rewriting plant DNA so the organism can physically emit its own light. Early attempts at creating living light were restricted by high maintenance. Startups like Glowy tried to illuminate spaces using bioluminescent marine bacteria, but these systems required constant life support, tanks of salt water, air pumps, and regular feedings of yeast. Because the loop was open, the technology remained a laboratory curiosity until the companies eventually ran out of funding. To create a commercially viable glowing plant, researchers knew they had to abandon marine bacteria entirely. They needed a biological system that could sustain itself without any external help. They found the genetic blueprint for that self-sustaining cycle inside a tropical glowing mushroom species called Neonothopanus nambi. By inserting the bioluminescent DNA from this fungus into the genome of ordinary vascular plants, scientists bypassed the maintenance requirements of bacteria. For the first time, the plants could produce light using their own internal chemistry. Shifting from external chemical extraction to internal genetic reprogramming allows the plant to generate a continuous, self-sustaining glow entirely on its own. The reason mushroom DNA works inside a leafy green plant comes down to a shared biochemical language. The metabolic pathway that makes the fungus glow fits directly into a plant's natural biology. Plants naturally produce caffeic acid. A fungal enzyme converts this into hispidin. Next, two enzymes oxidize hispidin, releasing a photon of green light and a waste molecule. Finally, a fourth enzyme recycles that waste back into caffeic acid, fueling a continuous, self-sustaining loop. Because it recycles its own internal fuel, this system requires zero batteries, requires no wiring, and no chemical sprays. To keep the light on, you just give the plant normal sunlight and water. By tapping directly into a metabolic stream the plant already possesses, the organism generates its own light simply by living. The very first generation of this technology was tested in tobacco plants. The loop worked, but the glow was faint. You needed specialized cameras in a pitch-black room just to see it. To fix this, a global team of scientists used artificial intelligence and directed evolution to study the fungal proteins. They identified specific mutations in the enzymes that sped up the metabolic cycle, eventually boosting the plant's light output by over 100 times. That algorithmic upgrade allowed the biotechnology firm Light Bio to create the firefly Petunia. By day, it looks like a standard white flower, but at night, the flowers and buds emit a continuous green glow. Before it could reach consumers, the U.S. Department of Agriculture ran a rigorous regulatory status review. They confirmed that the modified Petunia doesn't cross-pollinate with native flora and poses no plant pest risk. With that federal clearance, bioluminescence transitioned from a laboratory experiment into a product that can be safely grown in American homes. A glowing houseplant is a proof of concept. The underlying technology sets the stage for a shift toward programmable biological infrastructure. Because the intensity of the light is tied to the plant's metabolic rate, any changes in the organism's health cause the glow to fluctuate. The plant essentially broadcasts its internal state. Researchers are now taking this fungal bioluminescence cycle and linking it to specific stress-responsive genes inside major food crops. Imagine looking out over a field of corn at night. If the crops detect a fungal pathogen or experience a nitrogen deficiency, those specific stressors activate the luciferase genes, and the affected plants physically light up. This enables farmers to visually identify a stress response days or even weeks before any physical damage or wilting appears on the leaves. Engineering our crops to act as self-reporting biosensors offers a high-resolution window into crop health, moving farming toward a model of early, targeted intervention. Beyond agriculture, several startups are working to scale this fungal pathway to the municipal level, embedding this bioluminescence directly into the urban foliage. While LED streetlights provide the high-intensity light required for high-speed roads, biological illumination offers a lower-impact way to provide ambient light for park paths and residential walkways. A futuristic city park lined with glowing trees provides supplemental light that doesn't rely on the municipal power grid. These living sources provide a dim moonlight-like glow that illuminates pathways, while the trees actively pull carbon dioxide out of the atmosphere. We've spent centuries extracting useful chemicals out of plants to serve our needs. Now, we are writing code directly into their DNA, directing their natural metabolism to perform specific functional tasks. The glowing petunia on the windowsill is exactly what it looks like. The first functional example of a landscape where the infrastructure doesn't just sit there, it grows, reacts, and powers itself.